Hello everyone, my name is Xiaofang Wei. I prefer to be called Fiona. I'm from Petra, China, focused on research area of MUR, microbiology enhanced oil recovery. My presentation title is The Mechanism Study on Surfactant and Bio-surfactant Combination Flooding in EOR Technology. My presentation includes 4 parts. The first part, let's give a brief introduction of background. Currently, oil and gas are still the most significant reserve of all time, and its price will definitely be soaring when the regional or global issue happens. Oil and gas are still the most stable and reliable energy for security. As we know, the common surfactants used in EOR technology include chemical and bio-surfactant. Here we focus on the surfactant, Betaine, and glycolipid, rhammnolipid. Normally here, there are 2 kinds of chemical structure of rhammnolipid. They are monorhamnolipid and di-rhamnolipid. And the product of commercial is normally a mixture of rhammnolipid. Bio-surfactant has been used in EOR technology for a long time. The EOR effectiveness is not as good as expected, but its characters of environmental friendly, low CMC, huge range of pH and salinity, low IFT with oil have been accepted with most of the customers. On the contrary, the chemical surfactant is of good oil recovery performance with a kind of environmental pollution. In this study, the surfactant and bio-surfactant will be combined and evaluated. It is supposed to achieve a perfect surfactant agent with a better oil recovery performance and lower environmental impact. We also hope to probe the mechanism of surfactant and bio-surfactant combination flooding in EOR technology. Secondly, we'll talk about the material and the methods used in the experiment. Rhammnolipid is from a commercial product. In total, we bought 3 cans of rhammnolipids. They are purified monorhamnolipid, purified di-rhamnolipid, and a mixture of mono and di-rhamnolipid. The chemical betaine is synthesized by rhammnolipid. Oil sample is from the Yanchang Oil Field, Shanxi Province, China. It is a kind of sandstone with medium and low permeability reservoir. Temperature is from 15 to 63. Oil viscosity is less than 30. The equipment used in this experiment, IFT meter, is TS500C from USA. And the contact angle meter is OCA20 from Germany. And the surfactant configuration analysis was used by AFM made in broker. And the sample procedure was shown on this slide. Thirdly, we will talk about the third part, results and the conclusion. The IFT between rhammnolipid was measured with concentration from 0 to 2000 ppm. Compared to the monorhamnolipid, the di-rhamnolipid could reduce the IFT to extra low level, which is less than 10 to power minus 2. This means the di-rhamnolipid will be the key factor on IFT chains. The ratio between RHA and the betaine was selected by a lot of experimental data. The total concentration of 0.2% was fixed, both by the experimental data and the inside data. The ratio was selected 1:1 due to a stable low IFT data, which is less than 10 to power of minus 2. And this figure shows the IFT changes of combined surfactant and betaine only. The data confirmed the combined surfactant is quite good than the betaine only with relatively low concentration of betaine, which is 0.2%. Emulsification was also studied in this experiment. These pictures told us that the 3 cans of surfactant could mix with oil completely, but the mixture will be layered with time. Only the mixture with combined surfactant is stable. The contact angle was measured between oil and oleophilic rock. It is obvious that the spreading rate sequence as follows: di-rhamnolipid, betaine, monorhamnolipid, which means the oil separating ability by the combined surfactants may be mainly from the monorhamnolipid. We also hope to know the molecular configuration of the surfactant to probe the mechanism of the surfactant for EOR technology. The pictures of AFM was shown in figure 8 to 12. The concentration of the surfactant is a real concentration in the solution, which is 0.1% for each substance, and the mixture is 0.2%. The configuration of single molecule of monorhamnolipid could be seen clearly. It is not so close, which means maybe it is not be easy to form a shell. But for the rhammnolipid, the peak sharp is sharp but closed. This means it is more easy to form a shell due to intermolecular association. From the betaine, the configuration is fine and closed. This may be explained. The property of its interface is stable, but the combined of the surfactant picture is quite different. The betaine and monorhamnolipid is sharp and rocky. The betaine and the di-rhamnolipid is relatively flat, fine, and closed. This means the molecular configuration is quite different, but the detail of the configuration should be analyzed further. The conclusions have been drawn. The first, the oil separating ability of combined surfactant may be mainly from monorhamnolipid. Di-rhamnolipid could reduce the instant IFT value. The combined surfactant could reach to extra low IFT with a relatively low concentration of betaine, and the parameters of contact angle, emulsibility are same or higher level compared with betaine only, which means the environmental impact will be reduced greatly based on a similar or a higher EOR rate. And last, we talk about the way forward. In this experiment, we realize that the bio-surfactant plays the role of sacrificed reagent, which will lead to a better oil recovery performance. Most of the bio-surfactants stay in the formation instead of chemical surfactant, which will reduce the environmental impact greatly. Actually, the experiment is still continued. I reported the chilled data here. A lot future work should be carried out. We plan to further probe the mechanism of surfactant and the bio-surfactant combination flooding in EOR technology, and further analyze the molecular configuration detail of the rhammnolipid and the betaine, and the experiment of co-flooding in laboratory will also carried out recently. That's all. Thank you. Thank you for your attention.